Transgenesis is the introduction of some foreign gene into a host animal that is made by the intervention of a human being. Now, there are many applications of transgenesis, and there are many methods of making transgenic animals. We will discuss in this session what are the different methods of making transgenic animals, especially poultry, and what are their applications. So I think there are many students are there in this group. So I will touch upon the basics of molecular biology before going into the transgenic section. So to start with, you know the cell and the nucleus inside, and all the genome material is packed inside the nucleus of the cell. So this genetic material, the DNA, is packed in chromosomes. The number of chromosomes vary from species to species. And this genetic information is transferred from parents to offspring. So nucleus is the place where the genetic material is stored, and in chromosomes. So the quantity of the DNA present inside a cell, that is a single set of chromosomes, is called genome. So it varies from species to species. Again, the number of chromosomes varies. You know, chicken has 78 chromosomes. So human being has 23 pairs of chromosomes. And so this genome size is also different. In mammals, you see there are 3 billion base pairs of DNA. Now, what is this base pair? So, if you look at the structure of the chromosomes, chromosome is made up of the DNA, and it is packed with nucleosomes, tightly packed. And this DNA contains the genes. All the DNA is not genes. There are some non-coding regions also there. And this is on the right side. This is the structure of DNA, and this DNA is made up of the purines and pyrimidines, the bases, and one pair of base here is called the base pair. And the total length of the DNA, or the total size of the DNA in cattle, is around 3 billion base pairs. Whereas in case of chicken, it is just 1/3, around 1 billion base pairs. Now, this is the raw genome data. It looks like this. And all this A, C, G, T, all the 4 bases are occurring repeatedly. And then it runs without any punctuation or any spaces. And coming to the chicken genome. So, as I told you, it contains 1 billion base pairs, total genome size, and it contains 20,000 to 23,000 of genes. Now, the number of genes, it is almost similar to the cattle, even though the total genome size is less, 1/3, the number of genes is almost same. So how it is possible? It is possible because the genes are concentrated more in case of poultry. Most major part of the non-coding regions are not present in chicken. So the gene concentration is also very high, 4 to 5 genes in 1 megabase pair of the DNA. So less junk DNA, only 10% of repetitive DNA. These are all the specialities of chicken genome. So if you print these base pairs, ACGT into a book. So it will contain 200 volumes of books of 300 pages. You can imagine how much data is stored in a very tiny nucleus. So this information is available, is packed in all the cells. All the cells has this much of information, and if you unwound this DNA, it will runs to a mile length, 1 mile of length. So that much DNA is packed in the genome nucleus. So now, what is gene? So there are, I told you, around 20,000 to 24,000 genes are there in chicken. And this gene contains some coding region, that is open reading frame, and some regulatory sequence which is lying outside on both the side, promoter, enhancer. And this gene is translated, transcribed into mRNA, and then it is translated into the protein or polypeptide. And these polypeptides, they again, after post-transcriptional modification, they are responsible for all the functions or all the characters of individual. So this is the structure of gene, and it's nothing but the filament. DNA filament. There are different parts of the genes, and it is transcribed, and it gives a single stranded RNA. And after processing it, the mature transcript is formed, and then from this, the protein is synthesized. So this is the central dogma of the molecular biology, and this is how it works. So this is in a nutshell, from chromosomes to the end product, the protein. Now we'll see the animals shown on the screen. Some animals are glowing, some animals are not. See, these birds are glowing, and this is not. And this mouse is glowing, and this is not. Some colonies are glowing, but others are not. Why? Why it happens? I told you there are around 24,000 of genes are there, and all the animals, birds are have that genes. But these birds, the birds which are glowing, has some extra gene for glowing, which is introduced by human intervention into the birds. That's why they glow. So the gene which is introduced here is a green fluorescent protein gene, GFP gene, and which gives the fluorescence or glowing in the UV light. Now see the fish. The upper one is the wild fish, and the lower one is the transgenic fish of same age. See the size difference. Why it is happen? So there is some the growth hormone is switched on, like a switch, like a switched on mechanism is introduced into this genome of this fish. That's why it grows faster. And now, what is the transgenic chicken? It's carrying a recombinant DNA molecule, which is introduced by human intervention. And this gene is called the transgene, and the product produced by this gene is called transgenic product. Now, this is the history of the transgenic animals production. So it started in 1981 with first report by Gordon and Ruddle in mice by microinjection. Then in 1989 by Salter and Crittenden by virus-mediated transgenesis. It goes on, and many transgenic chickens have been reported later. And now the editing, gene editing, all recent developments in biotechnology has helped a lot in producing the transgenic birds. Now, what is the advantage of chicken to have it as a transgenic animal? You know, the chicken can produce around 300 eggs, more than 300 eggs. and more than 10% of that is protein, and again in protein, around 50% is albumin. So if you calculate, a chicken can produce around 20 kg of egg in a year, and it can produce around 1 kg of albumin. And if we could modify it genetically to get some medically important products, biologically important biomolecules from the birds. So it can produce more than, if even 1/4 of the albumin produced is modified like that, then 250 grams of modified recombinant protein. So that will be of a very high value. So that is the potential of the chicken. The other one is the egg is sealed in eggshell, so no contamination occurs, unlike if you produce the transgenic proteins in milk. The purification is a very difficult process. So it is another advantage for chicken. And also the cost of production is also, of course, less. Now, coming to the biofarming. So I will, so here, so I told the gene structure of gene, and if I can construct a gene with the regulatory sequence of the egg protein and a coding sequence of a foreign gene of therapeutically important. So this transgene, this construct, if I can insert in a chicken, so the foreign gene can be expressed in egg. So the protein which is produced in egg can be used for therapeutic purpose. So this is the basic of the biofarming. Now, so so not everything is good, but in avian transgenesis there are many difficulties, because the poultry is oviparous, and it produces an egg inside its body, and it lays, and the chick develops outside the body. And the egg formation is also different. The ovum is ovulated, and then it goes into the infundibulum, where the fertilization takes place within half an hour. The fertilization takes place in infundibulum. Then when the yolk passes down in the magnum, the albumin is secreted, 4 layers of albumin is secreted, and in uterus, uh, in isthmus, shell membrane is produced, and in uterus, shell is produced over there. So there are many layers deposited on the ovum. The other difficulty is even this ovum is embedded in a yolk material, so it is not transparent, so it is not visible for any manipulation. So these are the difficulties, and also the nuclear transfer is not possible in chicken, because for a nuclear transfer you have to manipulate the ovum, but as it is embedded in yolk, and again it is covered by many layers of egg, a single cell ovum is not accessible for manipulation in case of poultry. So there are some peculiarities in avian species. The birds are males are homogamous in chicken, and females are heterogamous, just opposite to human being. So here the sex is determined by female birds. And also the other one is the red blood cells of avian species are nucleated. So for molecular biologists, it is very easy to collect the DNA, to isolate DNA. All the cells in the blood are nucleated. And in birds, only left ovary and oviduct are functional. So these are all some peculiarity we have to keep in mind. So there are different methods of making transgenic animals or birds. One is virus-mediated gene transfer, where the virus, mainly the tumor-causing virus or oncogenic virus, are used as vector to carry the foreign gene into the genome of the host animal. The nuclear transfer is another method where the nucleus of the host ovum is removed or enucleated, and the new nucleus is placed inside, and the ovum is again allowed to grow. In case of microinjection, a few hundreds of copies of DNA is injected into the ovum before fertilization, before the fusion of both nucleus of male and female pronuclei. So few copies of foreign gene can integrate into the host genome. The other method is cell-based methods, mainly the embryonic stem cells and primordial germ cells. So these germ cells have to be collected from a donor embryo during very early stage of its development, like 3 days of its development. Then these cells can be cultured in labs, and again the cultured cells can be modified with the foreign gene. You can introduce the foreign gene in those cells. Again, these transgenic cells, primordial germ cell or embryonic stem cells, can be introduced into another host. So the host will become transgenic. So in this case, you can produce interspecies or intraspecies transgenic birds. And the other one is the testis-mediated gene transfer, where the spermatozoa produced from the testis is made transgenic, and the other one is sperm-mediated gene transfer, where the sperms are used as vector to carry the foreign gene into the ovum. So we'll see one by one. The first one is the virus-mediated gene transfer. So these oncogenic viruses have the ability to convert its RNA genome into DNA by reverse transcription process, and they have the ability to integrate their genome with the host genome. So if you make the virus particles which carry the foreign gene, so along with the integration of its own genome, it will also integrate the foreign gene also. So this is one method of making the transgenic animals, and it should be done in very early stage, and it will produce in the production of chimeras. So all the cells are not infected. If complete infection is not happening, then some cells will have the original host genome. The other cells will have the transgenic genome. So the birds come out or grow out of that zygote will be a chimera. It contains the cells of both the host and the transgenic cells also. And so these are the different viruses. All these viruses are the uh, oncogenic viruses, avian leucovirus, retrovirus, and reticuloendotheliosis virus and lentivirus. So there are many research workers. They used different the oncogenic viruses to act as a carrier of the foreign gene into the host. So even though retrovirus was very commonly used in earlier days, now people they go for lentivirus, which is more efficient in doing this job. So the other problem with virus-mediated gene transfer is it can carry only uh, a limited size of foreign gene, like 8 to 10 kb size only, and it will integrate its the trans gene in the host chromosome at random sites. So you cannot control the site of integration in this virus-mediated gene transfer. That is another problem. And also due to this, the foreign gene integrated may not act, may not expressed because of the silencing effect from the neighboring genes. The foreign gene we introduce may not express. So that is another problem with this virus-mediated gene transfer. And the other problem with the virus-mediated gene transfer is the infection. It is dangerous to the handling person or researcher also. It can cause the tumor in researcher also. So to avoid this, replication deficient virus can be used. There are two types of virus used. One is replication competent and the replication deficient. The replication competent virus, once you use, it will again replicate. So it is dangerous to the handler. Whereas the replication deficient form of the virus, it will integrate, it will do all the function of integration of foreign gene, everything, but it cannot replicate in the host cell. Thereby, it is safe compared to the replication competent virus. So this is one example by which the transgenic birds were used by Lindy virus. So these are the advantages and disadvantages I have already told. So this is another here this is the construct which is used, and it has a GFP protein. So the DNA construct, it has a GFP protein and the CMB gene also, and this construct is inserted into the blood vessels. Carrying the virus carrying this construct injected into the blood vessel, and the transgenic coil is produced. The same thing, the same picture on the left side. This is the under UV fluorescent microscope, and this is light microscope. You can see the difference. And has a big breakthrough through in 2012. Scientists from Roslin Institute, they have developed a chicken which is resistant to avian influenza, AA. They used this virus mediated gene transfer, the virus which carries the gene for short RNA genes. So these molecules, shRNA genes, which are introduced, which are integrated into the genome of the host, produces shRNA, and this interferes the multiplication of avian influenza virus inside the host cell. So this bird, even if it gets the disease, it cannot transmit the disease. So it was happened in 2012. And you might have seen this Dolly sheep in 1966. It made big news. And this is cloned from a donor sheep from a somatic cell. And this is just a replica of that donor. So how it is made? It is made by a nuclear transfer. So this is the second method of making transgenic animal. Here, this method cannot be employed in poultry because, as I explained, single cell ovum is not available for manipulation in case of poultry. So this method is not used in poultry. Just for academic interest, I am telling what is what. So here, this is the ovum of a recipient, and this is the somatic cell of a donor. From donor, the nucleus is taken, and from recipient, nucleus is taken out, and this donor nucleus is inserted into the ovum, and it grows into animal. Now, this is the nuclear donor. This is the ovum donor, and the offspring. This is a surrogate animal. So there are three animals involved. A somatic cell from the nuclear donor is taken, and the nucleus of that cell is introduced into the ovum of the ovum donor, and after that, after giving some stimulation to grow, this ovum with the nucleus of this animal will be uh, implanted in the surrogate animal, and it gives an individual which is the clone of X sheep. This is the clone of this sheep, and this is just a replica of its parent. So it contains all the genetic material what it donor has. So in normal fertilization, 50% of genes coming from mother and 50% from father. But this individual has all the genetic material comes 100% comes from one donor. So this is so this at this stage, these cells can be digested and separated. Each cell can grow into different embryo. So from a single cell, you can produce many clones of same animal. So these all the individuals in this picture are the clones of a particular donor. So it is 100% replica of its donor. So here, a donor and transgenic animal, and it's the progeny of the transgenic animal. So this is second generation. This is the first generation, and this is second generation clone. Now coming to the third method, micro injection. This method is very much useful or very much practiced in animals like mouse. It is very simple, mouse and rat, and where this micro injection station is needed for this, and where the embryo is holded with the holding pipette, and this is the injection needle containing the DNA, foreign DNA, few hundreds of copies of foreign DNA. This needle is pierced in the ovum, so it's a fertilized ovum, but the pro-nucleus of both male and female have not joined yet. It is fertilized, but the nucleus have not joined yet. So the foreign genes is introduced or injected in the pro-nuclear area. So it disturbs the nucleus and the genome there, and it will make some cuts, and the foreign gene we introduce may get integrated with during the repair process of the DNA. So foreign gene will get introduced in that, and the animal which grow out of this embryo will carry the foreign gene. There may be one copy or more than one copy of that gene in that animal. It was tried in chicken in 90s, but there are more reports after 1995. Only up to 1994, it was tried. It was tried. Again, here the problem is getting a single-celled ovum. But the scientists, Lau et al., they sacrificed the bird after around 2.5 hours of laying. They sacrificed the bird to harvest the ovum, next ovulated, which should be in the magnum, the upper portion of magnum, just fertilized. So that was used, and then they used a transgenic animal bird using with the LAC-Z gene, and they combined the viral mediated gene transfer. So here again, even you collect the single cell cycle after sacrificing the bird, because in case of bird, the fertilization takes place at the infundibulum, and then after 2 hours, the first division takes place in magnum, And after every 20 minutes, the division takes place, cell division takes place. So when the the uh, uh, the egg is laid, it contains around thousands of cells. It contains an embryo having thousands of cells. So to get a single cell zygote, you have to sacrifice the bird. You have to collect the ova from the magnum, and again, it is not possible for micro injection or injecting the foreign gene in the pronuclear area because it is not visible. It is embedded in the yolk. What this scientists did, they used virus. They infected the embryo with virus, so and they produced the chicken, uh, transgenic chicken. So this is another technique. It's a window technique. A portion of the shell is cut, and the virus carrying the foreign gene can be allowed to infect the growing embryo, and you will get the transgenic birds. So this is a report by Scott and Lewis. They used replication deficient lentivirus to produce transgenic birds. So I had opportunity to visit France, Nantes, where I underwent the injection of the micro injection of rat embryos. This is what I performed there. I was trained in transgenic animals there, so I practiced this technique, uh, micro injection in rats. So where in the lab where I work, they produce the transgenic rats very simply, just like a daily routine job there. Now the fourth method is cell-based technology. Uh, Here, three types of cells are used. One is blastodermal cell, the other one is embryonic stem cells or primordial germ cells. So the blastodermal cells arise at early stage of embryonic growth during stage 10. So it is around 1.5 days of incubation, very early stage of incubation. And at around 12 to 14 stage, that is this around 2 days of incubation, and the embryonic stem cells are there, and primordial germ cells are also arising. So these are the primordial germ cells. These cells you can see. So in initial stage, it will circulate in the blood. You can collect from the blood of a donor embryo, and so after some time, these cells will settle in the gonadal ridge, and it will form the gonad, either ovary or testis. So before it settles there, you have to collect the primordial germ cells, and the primordial germ cells you collected can be cultured in the laboratory, and where you can make all the modifications. You can introduce new gene, and the modified primordial germ cells can be introduced in the another host. So this is the nutshell. So collecting the primordial germ cells or blastodermal. This is blastodermal cells. Collecting blastodermal cell, culturing in the laboratory, and adding a transgene into that. So these are the transgenic cells, blue color. And by using some marker genes, you can select the transgenic cells, the blue ones. And these transgenic cells are introduced into a host, another embryo. And this embryo will develop into a chimera again, because it contains both the transgenic cells and its own cells. So both type of cells will be there, tissues will be there in the hatched out egg, or sorry, hatched out chick. So that's why we call it as chimera. So it can be used as a founder, and then by breeding a transgenic animal from this chimera. So during this culture in the laboratory, now this genome editing is also applied. So this talent and other methods of genome editing, it improves the efficiency of integration of the foreign gene into the host genome. So this I explained already. So here when we culture the cells in laboratory and when we insert the foreign gene, the insertion of foreign gene may be a homologous recombination or a random insertion. So homologous recombination means we have a modified gene, and the modified gene is inserted into its original gene, and the original gene is removed and this modified gene is inserted in the same place. So then it is called homologous recombination, and if it is somewhere else, insertion. So this homologous insertion and random insertion. Here, this is the vector or the foreign gene, or the homologous to the. This is the genome of the host. Genome of the host. So in genome of the host, sorry, this is the genome of the host. This is the genome of the host. You see the homologous areas of this genome of the host is made in the transgenic construct also, these blue areas, right? So by this recombination, this area instead of this area, this region will integrate here, and as a result, this genotype will be formed. Instead of A, we are removing this A and we are introducing a modified form of this gene, the same gene we are introducing here. So here, so instead of A, now it becomes a modified gene. This is called the homologous insertion or homologous recombination. On the other hand, if the foreign gene is inserted in some other place, some non-genic region, or in the area of some other gene, then it is called as the random insertion. So this type of genotype will occur. So when a native gene, a gene present is inactivated, now this A is inactivated with A star is introduced, then it is called, then this animal is called a knockout animal, because that gene originally present is removed or it is inactive. So then you can call this animal a knockout. Here, a new gene is introduced. So chicken or animal. So knockout technology, knock-in technology, and gene therapy. So if this gene is having some defects, then you can change that, you can modify that by molecular technology, and then a gene with a correct sequence can be inserted so that that disease can be prevented in the individuals or the progenies. So that if it is done for that purpose, then it is called as gene therapy. So these are the different methods comparative. Just I will go through only the embryonic stem cell. So embryonic stem cell is collected and cultured in lab, and the DNA is added, and it is again introduced into the host, and a resultant chicken is hatched out, and it is a chimera. Now from the chimera, how to develop a transgenic bird? So it has both the tissues of its own and the transgenic tissue. So first step, it is crossed with a normal individual. Then you will get some birds which are having having the foreign gene. Then you have to select those birds. Then again, one more. So this is now you got transgenic birds, but in lesser number. And also in first step, you will get only the one copy of gene. It will be heterozygous. Not, you will not get a homozygous, because only one parent is having that gene. So the next step, this transgenic bird, which is having only one copy, heterozygous, is crossed with again normal bird. You will get many individuals with transgene. And now, third step, when you cross a male and female of transgenic bird, you will get homozygous transgenic birds. So this is how from a chimera parent or base, you produce the transgenic birds. So this is the technology which is used in this type of making cell transfers, cell-based methods. So this is a donor egg where the embryo, so I told you 2 days old embryo is excised, and from this the uh, primordial germ cells are collected, and this is a surrogate shell. It's another shell, eggshell of another egg, in which the egg content of yet another embryo, 2 days, this is recipient embryo, is poured, and in this embryo, the cells collected and modified are inserted, and it is then covered with the cling film, and the incubation will be continued to hatch out a new chick, which will be a transgenic chick. So this technique, that is surrogate eggshell technology, is useful in developing the transgenic chicken. So this primordial germ cells can be used for producing many pharmaceutically important molecules, biomolecules, which can be used for treatment, therapy for human diseases. Now, the interspecific germline transmission. So I told you in cell-based methods, it may be interspecies or intraspecies. Now, interspecies means between two species. The same method, the collection of the stem cells will be done from one species, and it will be inserted into the host embryo of another species. Here in this, two species used are guinea fowl and chicken, and the stem cells of chicken is collected, and it is inserted into the guinea fowl, right? So the chimera heats were produced, young one of guinea fowl. Chimeric heats were produced, which carry the chicken cells also. So this chimeric guinea fowl will produce the sperms with chicken genome. So when this semen from this chimeric guinea fowl male is inseminated to a normal, ordinary female chicken, it produces chicks, right? The semen from guinea fowl is inseminated in hen, and the hen produces chicks. So this is called the interspecific germline transmission, interspecific chimeras. So these are different examples of duck and chicken chimera. So middle one. And again, this is a chimera, a duck, which carries chicken cells also. So the when the sperm is inseminated in this females, so it produces chicks. And also the major application of this method is the conservation of endangered birds. These Hobara bustards or the racing birds, which are endangered, and these birds, this type of birds can be conserved using a host interspecific chimera technology. Coming to next method, the testis mediated method here. and the DNA is injected into the testis. So this is a mouse, and this is the testis, and where the foreign gene is injected in the testis, and it is given uh, some electric shock-like thing, electrophoresis, to enhance the integration of foreign gene into the spermatogonia, and the sperms produced from those transgenic spermatogonia from the animal will be transgenic sperms. So that is one method, and so it is not a very popular method, and it is not very efficient also. And the other method is sperm-mediated gene transfer, where the sperm is collected from a donor, and the sperm is treated with some foreign gene in room temperature. So some sperms will take up the foreign gene, and the transgenic spermatozoa will be inseminated in normal hen, and the chicks produced will be transgenic. So semen collection, and the semen is processed because semen contains the DNAs in its seminal plasmas. Therefore, you have to wash. You have to wash the sperms to remove the seminal plasma. To separate 2-3 times washing is necessary. So, with the washed sperms, a DNA is added in a solution, and it is incubated at room temperature. So this naked DNA. So the naked DNA can be added, or the DNA mixed in liposome. So that method is called lipofaction. So liposome with the DNA, if you add, it will increase the efficiency of integration of the DNA into the sperm cells. And also you can use restriction enzymes also in this combination. So all these things, liposomes and restriction enzymes, will increase the efficiency of integration of foreign gene into the spermatozoa genome. So this is the restriction enzyme mediation. We call this restriction enzyme mediated integration, REMI. Or you can give some electrophoresis, some electric shock to enhance the integration of gene into the host genome. So we did some work in this in 2005 at IVRI. So my guide, late Dr. Deepak Sharma, under the guidance of him, I did some work on SMGT, sperm-mediated gene transfer. So here we took the 100 million processed or washed sperm cells, and we used the 10 microgram of exogenous DNA, and it is treated for 1 hour. So some cells will take the DNA. So after that, after incubation, then again we go for DNA treatment to destroy whatever DNA outside the cell. Only the DNA, the foreign DNA, which has gone inside the sperm cells, will be protected. Whatever extra DNA we have added here, if it is there, it will be destroyed. Then again, the DNA will be isolated from these sperm cells, and it was verified for the internalization of the DNA. So this is the vector DNA. The DNA we used both GFP gene as well as laxet gene. This is the vector, and this is the negative control, untransfected sperm's DNA. So there is no foreign gene there. This is the dot blot. So this technique is called dot blot. And in third, you find the nude DNA integration, transfection. What is this transfection? Transfection means the introduction of foreign gene into the cell by just incubation, right? So we use the term transformation in DNA cloning in E. coli cells, transformation, that is prokaryotic cells. When we use it in, do it in eukaryotic cells like sperm cells, it is called transfection. So, right. So this is made of the DNA of nude DNA transfected sperms, and these here you can see the little dark dots, which means here it is from the lipofected treatment, lipofaction. That means the lipofaction has improved the efficiency of internalization of DNA. This is a southern blot. Again, this is the vector DNA we used, and this is the positive control laxer gene as such. And this is the negative control, the DNA of non-transfected sperms, and this is the DNA of the nude DNA transfected sperms. So here you can see a faint band, and here the fifth one is the lipofected. You can see a strong band there in fifth. Now, the uses of this transgenic animal. There are many uses like gene farming to produce many pharmaceutically important molecules, and xenotransplantation, that is transplanting the organs to the human beings, even blood from species to species. And there are many industrial applications, many pharma companies can produce the pharmaceuticals and food and feed. The transgenic animal can have produce more product, egg, meat, or it can produce quality product, less cholesterol product, like that. So improved food quantity, it can contribute by increasing high feed efficiency, high rate of gain, overall body composition, increased breast yield, and body size, egg weight, right? And quality, by manipulation of biosynthetic pathways, it can reduce or improve fat composition in the carcass, overall body composition, lean or fat carcass, and less cholesterol, and the products, egg or meat containing some therapeutically valuable biomolecules in it, so that egg and meat can act as a medicine also. So biofarming, so we have discussed. So the achievements so far, there are many recombinant proteins are produced from these transgenic animals. There are many reports, and very few are efficient, or the amount of the transgenic product produced is significant, so that it can be used for commercial purpose. Originally, the yeast and bacterial are used for producing these monoclonal antibodies and other important therapeutic proteins. In 2009, the antithrombin 3. So it is the transgenic product produced from a goat milk, from a transgenic goat, which was approved by the FDA in 2009 for human purpose. So this medicine is available in the market, which is produced from a transgenic goat. So after 7 years, a transgenic chicken, which express the protein sebalipase alpha, which is used for the treatment of rare disease, lysosomal acid lipase deficiency. This is also approved now, which is also available. In 2017, there are 62 recombinant therapeutic proteins were approved by the CDER, Center for Drug Evaluation and Review, Center for Biological Evaluation and Review of FDA. So there are many products are coming from transgenic animals. So these are some of the products which have been produced. So this xenotransplantation. So there are heavy demand for the organs for transplantation. So many patients are dying without the organs. So the one thought is to get the organs like valves in heart or some parts of the organs which can be transmitted, which can be produced from other animals. So here, pig is think of because the size of the organs is comparatively to that of a human being. So transgenic, the research is going on to develop transgenic pigs which can donate organs for the human being. So this the transplantation of tissues from species to species was tried earlier from since 1905. But the animal, the human or the persons who received the transplant of other species died because of the histo incompatibility. So there are some genes which control that. So if you can make by transgenesis, if you switch off or knock down those genes in pig, so those organs can be used for human being for transplant. So these are some research findings. So in Roslin Institute and from USA, this I already explained in Roslin Institute, the chicken which is resistant for avian influenza, so antibodies, interferon beta 2, interferon beta 1A. Now the discussion is whether it can be used or whether it is safe to use. So there are many concerns which has to be addressed, like ethical, social concerns, medical concerns, environmental concerns, and economic concerns. Ethical concerns, whether it is ethics to make a transgenic animal, and economic concerns, it is costly. The methods are costly. It involves a lot of money and infrastructure, whether we have to go for that. Social concerns, many human rights groups and other religious groups, they are not for it. And medical concerns, whether it is safe, it has to be tested. Environmental concerns, if the transgenic animals escapes or go out of out from the custody, it may contaminate the wild animals. That is another concern. So these are the future challenges. So regulatory process are definitely required to regulate the newly produced transgenic animals, and to get the public confidence, they have to be enlightened with these techniques and other details. So it has now we have to take a decision whether to have it or not, or whether we have to have it for some kind of applications and not for others, by weighing its pros and cons. Uh, so this is an interesting story. Interesting story where the transgenic fishes were produced in 1980s. This is salmon fish, which can grow very fast and with very high feed efficiency, and which can grow in very harsh climates or harsh environment, polluted waters and all. This transgenic fish has insert from another species of fish, which switches on the growth hormone in these transgenic fishes. So it grows, even the environment is harsh, the temperature is very winter or it is non-conducive, the fish grows. So this technology was developed in Canada, and after many trials, FDA has approved this fish safe for human consumption in 2015. But many countries, especially US, was resistant to take this, and they banned that. Again, in 2019, they accepted because it is found safe. So this is about my presentation on Transgenesis. Thank you.
Okay, thank you, sir. Questions? One
All
from
right.
Dr. Sir, one from Dr. Soumya NP. Is it required to provide any special management care for the transgenic animals, and is it can we maintain them in field condition? That was the one question.
Okay. Transgenic animals, we are introducing a foreign gene, and we do not know how it behaves. So, as such, it does not require any special environment. But the transgenic animals, as I told, the fish, it is not allowed to grow in open ponds. So there are restrictions. It should be grown in restricted area. It should not be allowed to escape. And again, that transgenic, in particular, that fish, it is made triploid. So it cannot, even if it escapes, it cannot reproduce. So in that way, it is safe. So in field conditions, we are not having much transgenic animal in field. Even though it is produced by some laboratories, they are maintained in very high, strict safety conditions to avoid them to escape outside and contaminating. Right?
Okay, sir. Thank you. Another one from Dr. Vinita PG. What are all the legal concerns in development of these transgenic animals?
And it's again depends on country to country. We have worked in 2005. At those time, there was not much awareness of this transgenic. And later, one institute, I don't want to name, they did some work in transgenesis, but there was many controversies on that. And but now still, this transgenic chicken work is going on very vigorously in some institute, some ICAR institute. Legal concern, you have to get permission. You have to get the approved from the bodies, DBT and all. So there is a separate wing is there. So you have to get the approval of those.
Sir, okay, sir, thank you. Another one is like where we stand in Indian context, where we stand in production of this transgenic chicken, and which are all the things we should be looking for to transfer to or transfer in poultry, genes of interest.
So India, there is no transgenic chicken so far, to my knowledge. It is published. The published work I have not come across so far, but the work is going on very vigorously. So what is the other part of the question?
Sir, which are all the genes of interest for manipulation?
Genes of interest, I think I have explained adequately. So all the genes which have the therapeutic importance can be used.
Okay. Is there any difference in longevity of transgenic animal compared to a normal animal?
Longevity, there is no, it again depends which gene you are introducing. If you are introducing some, the gene you can introduce from same species, from one animal to other animal, or you can introduce from one species to other species, because the DNA is universal in structure, even though it is in cattle or in human being or in poultry, the structure of DNA is same, right? So it is possible to integrate a foreign gene from different species also, right? So when you do that, in that particular host, how the foreign gene works, right? Whether it has some health hazards in the host, that has to be studied. So whenever they make a foreign a transgenic animal, all these studies are done. Whether the animal is comfort or it is harmful to that animal itself because it is having a foreign gene. It is unnatural to that animal. So whether it is having any health hazards on that animal itself, it is all studied before it is the technology is accepted.
Okay, sir. Actually, we have some more questions that I think that will be answered in chat session because we are we have to start the next session by 11:30.
Okay, right.
Thank you, sir. Thank you for that excellent presentation. So now I invite Dr. Tirupati Venkatachalapati sir, who's chairing this session, to conclude the session. Sir, please.
Hello.
Yes, please, sir.
Yes, is it audible? Hello.
Yes,
Is it audible?
Yes, sir. We can hear you, sir.
Okay. So I thank Dr. R. Richard Churchill for his very nice presentation on transgenics. So I think he covered entire transgenics, not only in the poultry but also from the livestock also. So I hope this presentation will be useful and useful to many of you. So thank you very much. Richard Churchill, is it audible? Hello.
Yeah, yes, sir.
Hello.
Yes, sir. I'm hearing you, sir. I'm hearing you, sir.
Yes. Once again, on behalf of the organizer and behalf of the Kerala Veterinary and Animal Science University for your nice presentation. Thank you very much. Please.
Thanks a lot. Thank you for the opportunity. Thank you, sir.
Okay. On behalf of all organizers, I'm taking this opportunity to thank Dr. Richard Churchill, today's speaker, Dr. Tirupati Venkatachalapati sir, who chaired this session, and to all the participants. Thank you all. Have a nice day. And our next session, a role of poultry meat industry in boosting economy and Indian and US perspective, by Dr. Ranjit Ramanathan, will be starting at 11:30. All are registered to participate. Thank you. Thank you so much.
Thank you.